Welcome everyone to the second session of this wonderful celebration of Evelyn Witkin's life. We're going to be hearing, as Richard just said, about transcription-coupled repair. And yes, there's been huge progress and you're going to hear about it. But what I wanted to tell you about right now is how Evelyn came to this amazing finding. And what she wanted to do was really to quantitate the number of mutants after UV mutagenesis. So she wanted to plate cells under conditions where there would be low growth. She was using auxotrophs, and maybe there would be less of that particular amino acid as compared to when she plated it on rich media. What she found, to her surprise, was that when she plated the cells on this poor media, that she got almost no mutations compared to plating it on rich media. And that's what mutation frequency decline is. And her genius was to realize that likely on the poor media, there was some process that was correcting these mutations. So she went on to identify the gene that was responsible for that, which she called MFD, the MFD gene. And the others discovered that it was specifically involved in transcription-coupled repair, but Evelyn collaborated with Ashish Sankar to actually show in vitro that this MFD protein that she had identified genetically was in fact responsible for this process. And so with that brief introduction, I would like to go on to our speakers, many of whom I know well or are connected to in one way or another. And our first speaker is Dr. Seth Darst, the Jack Fishman professor, and who investigates the structure, function, and regulation of the bacterial transcription cycle, and is also, along with his wife Liz, a very good friend. I've enjoyed many evenings together with this family, and I should mention that Seth, like Evelyn, is not only a scientist, but a very accomplished pianist.